My name is Evan Spruit. I'm a group leader in Radboud University in Nijmegen. And this is one of the lectures that are being organized by the doctoral network, the EU doctoral network Commencell, which is on integrative synthetic cell biology, where we basically try to combine membrane-bound compartments, condensates, and metabolism and homeostasis. And we decided to organize an interdisciplinary lecture series in which different speakers from both within the Commencell network and outside share some of their expertise with everybody. So it's also my great pleasure to introduce the speaker of today, Professor Bert Poolman. Professor Bert Poolman is a professor in biochemistry at the University of Groningen, and is actually a very big name in the field of synthetic cell research. He is part of the basic build a synthetic cell consortium in the Netherlands, which is the consortium that aims to construct also a synthetic cell. And also part of the follow-up of this project, Evolve. Bert Poolman is also program director of the Center for Synthetic Biology in Groningen. And his expertise is mostly on membrane transporters, membrane enzymology, and how these membrane proteins help to establish and maintain homeostasis in synthetic cells. And I'm very excited that he is here today to tell us something about that. I would like to remind you before I give the floor to Bert that the lecture will be recorded, and we will place the recording on the YouTube channel afterwards. So please turn off your camera if you prefer not to be in view now, and then the lecture will be about 45 minutes. I would like to give the floor to Bert. We look forward to listening to you.
Thank you very much, Evan. So I'll immediately move to the next slide, if that is, yeah. So I was asked to present a lecture on metabolism and homeostasis, and I've given it the title Out of Equilibrium Reaction Networks and Homeostasis in a Selectively Open Environment. And I will explain these cryptic terms, out of equilibrium, homeostasis, and selectively open environment in the next few minutes. I would say metabolism is the chemistry of the cell, and if we specify it a little bit further, you could say it involves the reactions and interactions of all macromolecules and small molecules in a confined and crowded environment under mild conditions. And I would say homeostasis is chemistry under relatively constant conditions. And so what do I mean with mild? I will not say much about temperature. I assume that most biochemistry is run at between 20 and 40 degrees, even though life is possible at temperatures as high as, I think, up to 120 degrees. But what I will do is in the next couple of slides indicate sort of the physical chemical conditions that are typical of living cells, and also conditions that we ultimately try to implement in our synthetic cell. And so what I'm showing here is the case for a cell, and it is largely based on numbers that I've taken from E. coli. So a cell with a cytoplasmic volume of about 1 femtoliter. And if you take such a cell, then it is highly crowded and highly dynamic. And if you add up all the number of proteins and RNA molecules of a cell, then they occupy roughly 20% of the volume of a cell. And that is what we refer to as the excluded volume of the cell, or in a more qualitative term, we also call it macromolecular crowding. What are the implications of such a high crowding, and what are the implications of an excluded volume of 20%? So what I've done here is, for the sake of simplicity, I assume that all proteins are globular and have a similar size, and the size is indicated here. And if I put them in a box, then roughly this box represents a volume in which one protein occupies about 20% of the volume. And this means that on average, proteins are a distance apart that is smaller than their own size. And this is the condition under normal growth. However, cells are also often exposed to osmotic stress, and so the most extreme case is a form of hypertonic stress where cells are plasmolyzing, and so basically here all the macromolecules are sort of collapsing on top of each other, and in this case the distance between the molecules is zero, and this results in an excluded volume of about 50%. And so all the chemistry of a cell under normal conditions is taking place under these crowded or even slightly more crowded conditions. And what are the implications? So what I'm drawing here is in 2 dimension also a crowded environment in which there are several of these green particles. And what I'm now trying to do is I'm trying to add one more particle. This is the molecule in black. And as you can probably appreciate, the crowding is so high that this black particle does not fit in between these green particles at most places. And in fact, only the places that I have highlighted here in, pink, those are the places where this molecule could be sitting. However, if I now make this molecule smaller, and I've made it a lot smaller in this case, now suddenly all this pinkish space is available for this molecule. And so basically what I'm saying here is that a reduction in the volume of a macromolecule increases the entropy and thus decreases the free energy of it. And that can result in an enhancement of folding, that can result in an increased stability of macromolecules, it can result in condensing of DNA, oligomerization of proteins, and in some cases even an increase in the reaction rates. And here I'm illustrating this in 2 simple cartoons for the case of folding and oligomerization. So typically a high crowding favors the folded state, and the high crowding favors the higher oligomeric state compared to a low crowding. And what I mean with high crowding, that these effects, which I call entropic effects, typically occur significantly at excluded volume values of more than 0.1 or 10%. Well, I've just shown the case of a bacterial cell where the average crowding may be around 20%. In mammalian cells, it's probably lower, 5 to 6% lower, but still relatively crowded compared to other reaction environments that you may be used to in chemistry or in biochemistry. So these are entropic effects, and they favor folding, stability, oligomerization, and so you would say that these are all favorable effects. However, crowding can also have less favorable effects, and that is due to the fact that, well, proteins are of course dynamic. They can interact with each other, they can interact with other macromolecules, and the chance of interacting with other molecules is of course higher the higher the crowding is. But if you interact with another molecule, this can in fact lower the free energy of the denatured state. Or in other words, this may destabilize proteins. And I'm illustrating this here in an animation that is based on the molecular dynamics simulation study, where a subunit of pyruvate dehydrogenase is interacting with surrounding proteins, and you can see that gradually over time actually the green protein gets unfolded. Well, this is a computational study, but a group that has been pioneering the effects of soft interactions on on proteins under crowding conditions is the group of Gary Pilak, and he has studied such protein interaction by NMR, both in vitro but also in vivo. And so these interactions I would refer to as enthalpic effects, and these can be negative compared to the entropic effects that I was just showing that can be favorable for macromolecules. Well, what happens to a given macromolecule is very dependent on the type of molecule, and it is not easy to predict it on forehand. But this is, of course, clearly something that one can study in synthetic systems, which are less complex than the in vivo systems that, for instance, Gary Pilak and his group are studying. Well, as we now also know, a cell is not homogeneous in terms of its content. This is a cartoon representing E. coli, and here you see the nucleoid, and you can see that the polysomes are excluded from the nucleoid, aggregated proteins are excluded from the nucleoid, but then there are of course also several types of condensates present that either play a role in cell division or DNA replication, transcription, and so on. Condensates, of course, have been pioneered in mammalian cells, but it is also clear that in bacterial, but also other forms of life, they are actually rather prominent. And here you can also see an EM picture of an electron-dense part of the cell, which also reflects a condensate, in this case in an E. coli cell. Well, this slide, a lot of this slide, what is on this slide is of course the topic of also the Common Cell Consortium. So I've just shown what macromolecular crowding can do. So this is about the macromolecules, the proteins and the RNA. But a cell also has small molecules, which I refer to as the osmolytes. And the difference in the osmolyte concentration between the inside and the outside we refer to as the turgor or turgor pressure, and which can roughly be estimated in as indicated here in this equation. But if you then look at the turgor in a gram-negative bacterium like E. coli, the turgor can be in the order of 2 to 3 atmospheres. But in gram-positive bacteria, but also in yeast and fungi, but also in plants, it can actually be above 10 atmospheres. And to put these numbers in perspective, I'm comparing it with the pressure of a car tire, which is typically in the range of 2 atmospheres. And so even an E. coli pressure, an E. coli cell has already a higher pressure than a car tire under normal conditions. And this is rather impressive if you think that a cell not only needs to resist this pressure, but it also needs to be able to grow and divide under conditions that it maintains such a pressure. Well, it's not surprising perhaps that cells also control the internal osmolyte concentration, especially when they are living in an environment where the outside osmolyte concentration may change, and as a consequence also the turgor. And here I'm showing an example where we have a cell in which the arrows indicate the turgor pressure. But if we now expose it to hypertonic stress, the turgor will decrease, and ultimately the cell may plasmolyze. And certainly plasmolyzing conditions are detrimental for cells. The crowding becomes extremely high, but typically also cells do not grow under these conditions. And so what cells do when they are confronted with a lowering of the turgor or even plasmolysis, what they start accumulating are so-called compatible solutes. And an example of a compatible solute is glycine betaine, which is a quaternary ammonium compound. And these are molecules that can be accumulated to very high levels, even up to molar, without having negative effects on the metabolism of a cell. And as you can see here, this molecule, when it is accumulated to very high levels, a cell may eventually restore the turgor. Well, there are several proteins in biology known to do this. We mostly focus on this transport protein, which is an ATP-driven transporter that transports glycine betaine in. It can do it up to 1 molar concentrations or even more in bacterial cells. And this is a system that is gated by ionic strength. And that also makes sense if you think about the stress that is exposed to a cell. So a normal E. coli cell will have a low internal ionic strength under normal conditions. But when we osmotically stress it, the cell shrinks and the internal ionic strength will go up. And so under these conditions, higher internal ionic strength, this system gets activated, starts pumping this molecule in. Well, for many years we thought that this was the only regulatory mechanism until 7-8 years ago we discovered that actually if you don't shut down this system in time, it starts accumulating so much glycine betaine that the cell may actually lyse. And what we now know is that there is a second messenger, which is known as a cyclic di-AMP, that can actually shut off the system at different values of ionic strength and thereby prevent unconstrained accumulation and lysis of the cell. I will come back to this molecule later as we are already implementing it in the construction of our synthetic cell. And so you could say what I'm showing here is part of volume homeostasis, but indirectly you could also look at it as being crowding homeostasis. So back to this slide. So from proteins to osmolytes we now go to ions, and ions are also part of the osmolyte concentration. And the most abundant ion in most cells is potassium. And then of course there should also be suitable counter ions. And if you take the case of an E. coli, then the internal ionic strength is about 0.2 molar. Ionic strength can differ between different organisms. In mammalian cells it's probably lower, in gram-positives it's probably higher, but for now we take it as something around 0.2 molar. Then if we zoom into protons, most forms of life maintain an internal pH which is between 7 and 7.5. And I've taken here the case of an internal pH of 7.2. And if you would do that for a cell with a volume of 1 femtoliter, then there are roughly only about 10 free protons in the cytoplasm. But the cell also has buffering compounds, and so a typical cell may have in the order of 100 millimolar of inorganic plus organic phosphate, and this corresponds to roughly 10 million protons, or a buffering capacity that allows about 10 million protons to be buffered, which may look a lot, but then if you think about the rate of respiration of a cell, so the rate at which protons are pumped out of the cell, that is in the order of 1 million to 10 million per second. And you can already easily see that such a buffering capacity would not be sufficient to maintain the pH around 7.2 for a very long period of time. And so there is a whole lot of work known on membrane proteins, mechanisms involved in pH homeostasis. I was a little bit lazy yesterday, and so what I did is I phrased a few questions to an AI program and asked it, can you generate an image of how pH homeostasis could look like, or pH and ion homeostasis? And basically this is summarized in this cartoon. So there are buffering molecules. There are, of course, the proton pumps, like the respiratory chains that pump out protons. But then, of course, we need various types of transport proteins, sodium proton antiporters, potassium proton antiporters, that regulate the internal ion concentration, including the internal pH. Then there are, of course, molecules that sense pH and adjust metabolism if needed. And then some organisms even have special acid resistance mechanisms. I will not go much further into detail on this, although in the remaining of my presentation I will show a few examples of where we have already some basic pH homeostasis working. And so what we try to do in my group and within the consortia that Evan already mentioned, we try to implement physical homeostasis in synthetic cells and study the impact of, for instance, crowding, pH, ionic strength, and so on metabolism, or you could say on enzymatic reaction networks. So now a brief introduction on what enzymes do. What you see here is a chart that you may be familiar with. It shows part of the biochemistry of a cell. You may actually recognize here part of the glycolytic pathway, and you may here recognize the citric acid cycle. Well, you see all sorts of intermediates, and in blue you see the names of enzymes. And what do enzymes do? They catalyze reactions. But compared to catalysts in chemistry, enzymes also have the possibility to couple reactions, and for instance to couple unfavorable and favorable reactions. And here I'm illustrating this for the case that we want to phosphorylate glucose and form glucose 6-phosphate. This is energetically an unfavorable reaction. But then in parallel we have a reaction where we hydrolyze ATP to ADP. And if we now combine the two, we couple the two, then basically we have the reaction that is carried out by hexokinase. And so basically the hydrolysis of ATP in fact drives the phosphorylation of the sugar. Well, we are studying this type of systems in synthetic cells, and what I hope to illustrate is a couple of observations that we have made on what the effects of confinement are, that we can have futile reactions that you would not immediately recognize in cellular systems, that we can have nonlinear behavior, that we can have enzymes that are promiscuous, we can have homeostasis effects, and we sometimes see emergent properties of even very simple reaction networks. And so the questions that we are asking in my group: what are the simplest reaction networks for a desired function? What are the requirements for such a system in confinement? What are the challenges for coupling of different reaction networks? And how can we achieve homeostasis? And so now a few of the terms that I had on my first slide with the title. And I would say the out of equilibrium operation and homeostasis are probably hallmarks of all forms of life. And with out of equilibrium operation, I mean that a cell needs to be selectively open such that there is a continuous supply of substrate and removal of products. And the intermediates then can be recycled. And I'm illustrating this here. So these are the substrates that are entering a cell. Here is the reaction network, and here is a product, and the product needs to go out. Well, many people in this field, in synthetic cell, study this in vesicles, but they are typically closed systems. And so what we are doing, we are implementing transport proteins that can either do this, or do both. And a great group of proteins are the so-called antipore systems that are relatively simple membrane proteins, but they can exchange the substrate for the product. And so if you have a reaction with a substrate and you have stoichiometrically coupled the excretion of the product, you also ensure that the product will not accumulate in the cell. And I will show a couple of examples later on in my lecture. Then homeostasis, and I would say this involves the very different and the very many mechanisms that allow a cell to recover from perturbations. And what I've indicated here on the y-axis is the parameter that can be the internal pH, ionic strength, volume, redox state, whatever. And so if over time the system is perturbed such that, let's say, the pH has dropped to too low a value, then a cell needs to sense this, needs to respond to it, such that over a relatively short period of time it allows the pH to increase again, such that the internal pH is back in what I would call the homeostasis window. And so this and these type of systems we are trying to study in our synthetic cell endeavors. And so what I'm now going to illustrate are an example of a vesicle system in which we make ATP, where we recycle ATP, a system where we in one vesicle make ATP, transport it out, bring it to another vesicle, and then the ATP goes back in the first vesicle. We've also used this cross-feeding to let a second vesicle grow, in this case by membrane expansion. I won't have time to show this. And at the end, I may briefly mention what we are doing here by coupling different modules, and in this case by integrating vesicle systems with a given function inside a bigger vesicle system, and then basically create systems that akin what eukaryotic cells do, that have organelles performing subsets of functions. But I will focus mostly on ATP production, recycling, and cross-feeding today. So ATP production, and we try to do as simple as possible, and that is not by mimicking how our mitochondria do it, because this is of course a beautiful system, the respiratory chain that pumps protons into the intermembrane space and uses the proton gradient to electrochemical proton gradient to make ATP. But this system is way too complicated and involves more than 100 proteins. The simplest system that we could think of for making ATP is this one, in which we are deaminating arginine, we're then forming citrulline. Citrulline is then converted to ornithine and carbonyl phosphate. Ornithine is now a product that leaves the cell via the same transporter as arginine is using to enter the cell. And so this is a strict coupling. Citrulline plus phosphate gives carbonyl phosphate, which is a high energy intermediate, which allows ATP to be made. Then we form carbamate, which is chemically unstable and decomposes into CO2 and ammonia. And in later slides, I will show this pathway in this way. So we have the reaction network, we have an antiporter taking care for substrate uptake and product excretion, and we also form CO2 and ammonia, which leaves the vesicle system by passive diffusion. But as I will illustrate later, this has also consequences for the operation of the system. And then, so per arginine, we form one molecule of ATP. So some features, some basic biochemistry. So the first reaction has an equilibrium constant of about 1 million. So that means that the equilibrium is highly in favor of citrulline formation from arginine. But then if we take the second enzyme, the B enzyme, it has an equilibrium constant in the opposite direction, and so this is a highly unfavorable reaction. In fact, this reaction we also run in our body, and it actually runs in the opposite direction, in the favorable direction towards making of citrulline. But here we need it in the unfavorable direction to form carboxyl phosphate. Well, this reaction is somewhat favorable, has an equilibrium constant of 5, and then here we have the spontaneous hydrolysis of carbamate into CO2. Well, these are equilibrium constants. Of course, you can then calculate also what the Gibbs free energy change under standard conditions is. And you can see this is the first reaction has a negative Gibbs free energy change, the second one a positive one, this one a slightly negative one, but together with the carbamate to CO2, the overall process has a delta G 0 under standard conditions of a little bit more than minus 30 kilojoules per mole. And so in principle, that should allow the formation of ATP. However, if you take these three enzymes and you put them in a test tube and you provide them with, let's say, 10 or 20 millimolar of arginine, you barely form any ATP. And we also know now why. And so, for instance, if we take, there are a number of reasons, but one of the reasons is that if you take the first enzyme, ARC-A, it uses arginine as a substrate, but at higher concentrations, it actually becomes inhibited by arginine. And so this is one of the reasons that this enzymatic network works very poorly in solution. However, it works very well if we put it in a vesicle system in which the arginine, which is illustrated here as the red square, is taken up, but immediately converted into an intermediate of the pathway, and also the end product of the pathway is immediately excreted, such that the internal arginine and ornithine concentration are actually kept relatively low. And under these conditions, the system is very well capable of making of ATP. One example of the performance of the pathway. And so if you look here at the top, if you look at the overall reaction equation, then from one arginine plus one phosphate and one ADP and 3 protons, we form ornithine plus ATP plus 2 ammonia and CO2. So we are consuming protons. And so what you would expect is that the internal pH should go up because we are consuming protons. But as you can see, the pH goes up for a short period of time, but then actually drops, and actually the inside starts acidifying. So what is going on? Well, we realized immediately that, of course, the pathway produces ammonia, not only here, but also here. And we also know that although ammonium ions are formed, and even though the equilibrium is in this direction, ammonia can actually leave the cell passively at a very high rate. And if that is happening, it leaves a proton behind. And we can see that here in a stop flow kinetic experiment. And I think the only thing you need to do is to look at this orange line. In this case, what we do is we add ammonium chloride to the vesicles, so basically the opposite direction as the ammonia is moving here. And what you can see is that the vesicles rapidly increase their internal pH on a time scale of, let's say, milliseconds. And that is because the ammonia is diffusing so quickly. And so even though we may be forming this on the inside, and because we have this equilibrium, ammonia will diffuse out rapidly and will diffuse into a very large external medium, and this will cause the production of a proton. Well, we have this happening here as well. We also realized that CO2 may also contribute to the acidification. But if you would add up the protons, here one, and here another one, you would at best have a situation where the internal pH remains constant. And then we realized that what we are doing here is we are having a system in which we are making ATP, and over time we are running out of ADP. And so I'm finishing this now. And what may actually then happen is that if ADP becomes limiting, then more and more arginine may be converted into citrulline. And if we are forming way more citrulline than that we are converting arginine into ornithine and the production of ATP, then we would be able to acidify the inside of the vesicles. And in fact, this is what we see. Under conditions that ADP is limiting, we form way more citrulline than we form ornithine. And basically, we have created a futile cycle in which arginine is futile hydrolyzed to citrulline, and actually can also leave via the same antiporter as ornithine is leaving. And so this futile cycle now explains the acidification of the inside volume of the vesicles. But then what you would also predict that there would be less futile hydrolysis if we would couple the production of ATP to the consumption of ATP. And here what we have done is in the vesicles with this pathway we have co-reconstituted this protein that is involved in volume regulation. And so this is a transport protein that pumps in glycine betaine and it consumes ATP. So the ATP that is formed from the deamination of arginine. And what you can then already immediately see, this was one of the first experiments not yet optimized that we performed, that when we have this transporter with a substrate present and we consume ATP, we already can keep the internal pH more constant than we can without this reaction. And you could say we already have a little bit of pH homeostasis by coupling the production and the consumption of ATP. I'm going to show you a few more things of what we have done in this system. And so I already mentioned that this transporter is involved in volume regulation. And so if we reconstitute it in vesicles, and we make it in vesicles in which the internal ionic strength is low, then the system is inactive. But what we now can do is we can osmotically upshift the vesicles such that they shrink. In fact, they become invaginated. And now, even though the total amount of ions has not changed because the volume has decreased, the internal ionic strength has gone up. And if we now look at the activity of the transporter, you can see at low ionic strength it's inactive. If we perform this upshift, the transporter is active. If we then shift back, the transporter is shut off. And if we then shift up again, the transporter is active. Well, if we let this transporter go for a long period of time, then we can actually even restore the volume of these vesicles into something that looks like this. But look at this time scale. So this is seconds. And so we've also run the system for hours. And so now this is the time scale of hours. And what you can see is that, and you can also see that the amounts of glycine betaine that are taken up are almost 2 orders of magnitude higher. And under the conditions that are illustrated here by the red line, the system is okayish, but you can see with the blue line, we go actually through a peak, through a maximum, and then the levels go down. And what we are seeing here is actually that part of our synthetic cells are lysing. And so even though we have the ionic strength gating working in these vesicles, we do not yet have the cyclic di-AMP control to prevent these vesicles from lysing. And so this is one of the things that we hope to implement in the future. So then we said, and I'm now checking my time, I think I still have about 10 minutes. So now we have the same system again, but then we said, well, can we also use it as an organelle, produce ATP and excrete the ATP? And for this we use the mitochondrial ATP-ADP carrier, which is again an antiporter. And then what we observed was that, and now we are measuring inside the vesicles, we are measuring the ATP-ADP ratio, so basically a measure of the ATP that is formed. And what you can see here is, if you look at the light blue curve, we are forming ATP, but the ATP-ADP ratio goes up, but then also drops over time. However, if we now add ADP at a given moment, then we see a transient drop, but then the system restores, and then it remains constant for hours. And so basically, the ATP-ADP ratio can be kept more constant in this system when we are removing ATP from the system than when we let the system run out of ADP. And then when we were studying this further, we were looking at how much ADP we needed, and so this is under slightly different conditions, but here you can see if we let the system run without ADP, we almost get a complete drop in the ATP-ADP ratio. If we let the system run with half a molar of ADP, we go to a stable state, a higher ATP-ADP ratio, even though we are taking ATP out of the system. But if we are somewhere in between, we see an enormous variation in the data. And so the error bars that you see here basically reflect the variation between several independent experiments when the outside ADP concentration was 0.1 millimolar. And so the question was, what's going on? Is this real or is this an experimental artifact? So we then started studying this enzyme, which is a kinase that forms ATP. And so the kinase has a substrate carbonyl phosphate plus magnesium ADP, and then here the products are formed. But we also found what this enzyme can do. It can not only use ADP, but it can also use magnesium ATP, and what it is then forming is actually adenosine tetraphosphate. Yeah, and so basically this is an ATP with an extra phosphate. Well, you could say, well, is this something odd of this enzyme? Well, a very well studied enzyme of such as pyruvate kinase, we see the same. Yeah, and so if the ATP-ADP ratio is somewhat off, you can actually start forming these types of molecules. Something we completely had not anticipated. And to be honest, before we started doing this work, I had never heard of this molecule. Also, something that was not in the literature and that we saw was that even though the enzyme has a hyperbolic dependence on the carbonyl phosphate concentration, the dependence on ADP is actually sigmoidal. And then including in our analysis so the formation of adenosine tetraphosphate, the fact that carbonyl phosphate is not super stable, this sigmoidal dependence that led Jan Borges Boreto, a postdoc, to make a mathematical model which in fact can very nicely describe the experimental observations that we are having. So this is the case where we have no ADP on the outside and the system produces ATP, but then the ATP-ADP ratio drops. And this is the case where we have roughly 0.1 millimolar of ADP on the outside, and so we have the enormous variation in the data. And so basically what we think is going on, many enzyme systems behave in a nonlinear manner, and they can become bistable, and that basically means that at a given driving factor, in this case the magnesium concentration, the system can be either in a low output state, so a low ATP-ADP ratio, or a high output state. And so basically, if I would go back to this previous graph, then basically without ADP, the system goes to a stable state, which is what we are seeing here. With 0.5 millimolar ADP, the system goes to a stable state, that we see here. And with 0.1 millimolar, the system can either go this way or that way. And since we always have, let's say, slight experimental variations, this is why we are actually observing this enormous variation. And so basically, here we are actually observing the bistability of this reaction network. And so the take-home message would be from here: we have futile hydrolysis of arginine, which can also lead to an internal acidification, and depending on the conditions, we can have side reactions such as the formation of adenosine tetraphosphate. And so the cooperativity of the kinase, together with the promiscuity of the enzyme for adenosine nucleotides, this can actually lead to bistability. And so what have we learned so far? So for a well-functioning reaction network, the produced ATP must also be consumed by another process, and this minimizes the futile reactions and enables already partial homeostasis, as we have seen. I don't have time to present it all, but basically a metabolic reaction network operates as a balanced assembly chain, like what you're seeing here. If I would take this guy out, then the system would no longer work, and that is, or if this guy would start working a little bit slower than the other guys would, the system would no longer be working. And this is precisely what we see in our analysis of simple reaction networks in synthetic cells. We also find new properties, new behavior, new reactions, and we can actually even see that reaction networks can become bistable. So then we said, well, okay, can we take this system one step further? And now we don't consume the ATP by just taking it out, but let it take up again by another vesicle, and then cycle the ADP back to the first vesicle. And this is shown here. So now we have the vesicles producing ATP, and here we have another vesicle consuming the ATP, but then delivering the ADP back to the first vesicle. And then also in this example, we're using the same ATP binding cassette transporter, and now this system is pumping glycine betaine into these vesicles, which is dependent of ATP production in these vesicles. And this is how it looks like. So the vesicles A are the ATP source, the vesicles B are the ATP sink. And this is one of these experiments which I expected may work, but actually this is one of the experiments that worked beautifully. And basically what we are seeing here is the leveling off of transport is because all the glycine betaine has been taken up by these vesicles. Yeah, and so here we would say we have a system of ATP cross-feeding between the vesicles, and you could see this as an example of syntrophy. You could now ask the question, is the system that you've developed so far, is it fully out of equilibrium? Well, it's a little bit more complicated than I've said so far. So this is the system making and excreting ATP. But as you probably all know, ATP has 4 negative charges, ADP has 3. The transport of ATP out and ADP in generates a membrane potential inside positive. This membrane potential inhibits the export of ATP. And you can see that also reflected in the ATP-ADP ratio we can actually stimulate the pathway if we dissipate the membrane potential. But if we don't do that, actually the pathway is inhibited because this transporter is inhibited. Also, if we transport ATP out and ADP back in, we deplete the internal phosphate pool. And so we fix that by using another carrier, which is the phosphate carrier, which actually brings in the phosphate again. And what you can now see, if we now have a system that also has the phosphate uptake, we even see higher levels of ATP than without. But again, the system is very sensitive to the membrane potential. Well, here I'm only highlighting the sensitivity of this system to the membrane potential. Indirectly, this system is also dependent of the membrane potential. And so building such a system makes you realize that things can sometimes be more complicated than you may have anticipated on forehand. And so the take-home message at this point is that we have a simple reaction network, yet we already have complex behavior. This I already mentioned, but now we see that charge translocation by antiporters leads to self-inhibition, and this is a way, in fact, that we are now implementing to study the role of the membrane potential in regulating the metabolism. We can do cross-feeding, so we can transfer building blocks between vesicles. I've shown it for the example where we were looking at the transport of glycine betaine, but together with a group of Martin Exterkate and Arnold Driessen, we've also used it to look at lipid synthesis, but I don't have time to show that. Yeah, and so this is one of the things that we are working on right now, encapsulating these ATP and other types of vesicles inside bigger ones. We convert the smaller vesicles into giant vesicles, and then we can trap them in microfluidic devices, and then we can monitor reactions in bigger cells directly under the microscope. But since I'm running out of time, I'll stop here with my lecture, but not before thanking my group. So this was a barbecue last summer together with the group of Dirk Slotboom, and this was in September on a retreat with the group of Petra, and I think some of you who are now listening were also present here, and thank you.